Good afternoon. Can you hear me? Now you can. I am Shirley Jackson. I'm president of Rensselaer Polytechnic Institute, and I'm your moderator this afternoon for our panel where we will be considering pandemics and big data with the intent to disrupt transmissible diseases. This panel will proceed in a two-part fashion where I'm going to actually begin the conversation from the broader perspective, and then we will transition to a more focused discussion among the experts that will be chaired by my colleague, Dr. Jonathan Dordick. And then we will open it to questions from the audience. Now, as we consider the fourth industrial revolution, which has been at the root of this annual meeting of New Champions, where we're experiencing a remarkable moment of convergence between the digital, the physical, and the biological worlds, it is important that we consider other connections as well. Because if we look at the great global challenges faced by humanity, including a changing climate, our food, water, and energy security, the need for sustainable infrastructure, our national and global security, and in the subject of this afternoon's discussion, human health and the mitigation of disease, it is clear that these challenges are interwoven, and they leave us subject to intersecting vulnerabilities with cascading consequences. Now, as far as pandemics are concerned, a complex chain of causality can affect the velocity and speed of pathogens through the two principal pathways of transmission, the bacterial pathway and the viral pathway, which we will discuss this afternoon, even as emerging diseases can in turn provoke widespread political, social, and economic upheavals far beyond what the enormous cost may be in human lives. Now, in the case of the recent Ebola outbreak, the nation of Liberia was especially hard hit because a convergence of factors that included cultural traditions in which relatives wash, dress, and bury their lost loved ones and contract the virus in the process, and as well as distrust of the government and its recommendations for stopping the spread of the disease. These were the complicating factors, especially after, in fact, a quarantine caused unrest in the city of Monrovia. Now, the emergence of the Zika virus as a serious public health threat offers us another important and example of intersecting vulnerabilities with cascading consequences. Now, recent outbreaks, in fact, have helped us to determine that this poorly understood virus causes microcephaly and brain damage in fetuses, and Guillain-Barré syndrome in adults, an autoimmune attack on the peripheral nerve system. Now, while the virus may have acquired a new virulence, the allocation of public resources also factors into these outbreaks. In fact, the particularly opportunistic mosquito species that transmit Zika actually nearly had been eliminated from the Americas in the 1960s through large-scale, systematic mosquito control programs. But then complacency set in, and control programs languished. As a result, not only is Zika now a serious threat, but the global incidence of dengue, which is carried by the same mosquito and which is a much more deadly disease as far as we know, has increased 30-fold over the last half century. Now, adding to the complexity of the medical and epidemiological challenges, Zika is not merely a vector-borne virus, but also communicable from person to person. At least that is the way it appears today. Although such transmission appears to be rare and is, as of now, poorly really understood. Now, downstream, the political and social issues associated with a potential pandemic include the fact that, of course, the 2016 Olympic Games will be held in Brazil, which have been a center of the Zika outbreak, potentially allowing some 500,000 foreign tourists to spread the virus around the globe. Now, equally complex is containing the risk of pandemics caused by the other principal pathway of transmission, bacteria. Given the rise of antibiotic resistance and a chain of causation that includes livestock farming as well as medical practices. In other words, such intersecting vulnerabilities cannot be understood from a single point of view, and they cannot be eliminated with a single pill. Fortunately, the fourth industrial revolution is giving us remarkable new tools to understand, assess, and hopefully prevent potential pandemics. And these include the rise of big data and the Internet of Things, which are bringing new sources of information into the challenge of transmissible diseases. And they include new tools to understand biological systems, which can now be seen as masses of data and allow us even to engineer such systems. So here to help us to consider the ways that the new computational and biological tools to prevent pandemics are three distinguished panelists. I'm so pleased that we have with us, to my far left, Dr. Suzan Murray, who is the head of the Smithsonian Institution's Wildlife Health Program, which addresses animal diseases and conservation medicine. Now, a doctor of veterinary medicine and a diplomate of the American College of Zoological Medicine, she is a principal investigator for the Smithsonian in the USAID program on emerging pandemic threats. And since I'm on the Board of Regents of the Smithsonian, I'm delighted and honored to be here with Dr. Murray. I'm also pleased to welcome one of our young faculty who is here as a young global scientist, Dr. Ji Heng, or we call her Dr. Heng Ji, who is the Edward P. Hamilton Development Chair Associate Professor in the Department of Computer Science at Rensselaer Polytechnic Institute. Now, her research focuses on information extraction on a massive scale by computer systems, using natural language processing across multiple languages, including non-mathematical sources that range from published medical and scientific papers to the information to be gleaned on social media. Appropriately enough, her laboratory at Rensselaer is called the Blender Lab. And of course, I welcome Dr. Jonathan Dordick, who is the Vice President for Research at Rensselaer Polytechnic Institute, and he is our Howard P. Isermann Professor of Chemical and Biological Engineering. Dr. Dordick's research focuses on drug discovery, biocatalysis, and radically new strategies to combat diseases and pathogens, including those focused on antibiotic-resistant bacteria. So let us begin with Dr. Murray, who is on the front lines of emerging and, shall we say, re-emerging diseases, identifying dangerous pathogens affecting animals that could potentially and often do leap to humans. In fact, given that an estimated 75% of new human diseases are zoological in origin or zoonotic, including the worrisome H5N1 avian influenza. Dr. Murray, can you say, what concerns you most? And then what policies can governments establish to minimize the risk of potential pandemics? And then where do you need science and new techniques to help you?
Well, first of all, thank you for the introduction. I think what concerns me the most is also something that presents us the greatest opportunity, and that, as you had noted, that about 75% of the emerging infectious diseases come from wildlife first and then pass into the human population. And currently, we're in a situation where often we find ourselves waiting till a number of people get very ill and die, and then trying to figure out what the pathogen is, and then track backwards to figure out what's the pathogen, where is the host, the reservoir species, and then from there elucidate the transmission pathways, and then try to find ways to interrupt that. So by the time we've done that, we've lost lots of, tens of thousands of human lives in some situations. The reason that this also gives me a lot of hope is that we have a tremendous opportunity to do something. And that's one of the, we're fortunate enough to participate in one of those USAID Emerging Pandemic Threats program that does pretty much just that. Using modeling, it has identified the hotspots around the world where the next emerging infectious diseases are most likely to come from, the species from which those are most likely to emerge. And then we have teams that conduct or develop wildlife surveillance protocols in each of these nations, laboratory capacity. So with the idea of developing an understanding of what viruses are there ahead of time, and then understanding the pathways, so if people do get ill, especially at those high-risk interfaces between wildlife and humans, that we'll have some of that information ahead of time. So my concern is that we're not quite there yet, but the opportunity, I think, is so huge, and I think we've really learned through a lot of these, the last 10 years, how much physicians, veterinarians, virologists, biologists can do when we all work together and share that information up front.
You know, there's a lot of discussion, and you know, about the Internet of Things here in the Fourth Industrial Revolution, where there's a lot of censoring of the world. There's a lot of data. Yeah. Are there sentinels in terms of monitoring animals globally that can help you have a sense of the propagation of disease and how it may be changing species to species? Sentinels
meaning different species that we should be watching? Or?
Well, or how what affects animals in one place may show up in another, and may show up then with a somewhat different presentation, and whether that tells you something about what's happening with the evolution of the disease.
Oh, that's a little bit of a hard one because things are changing so much right now, and things change. The risk factors change from country to country and species to species. And now that we see a lot of the temperature or a lot of climate change, we also see vectors of disease moving into new areas. So mosquitoes and ticks now overwinter in places that they hadn't before. So I would say that there are so many things that continue to change, that the more we can stay on top of things and investigate up front and learn, and then share that information openly to everyone, the more successful we'll be.
Now, you've actually spoken about the behavioral factors in terms of disease propagation.
Right. There's a lot of things that people do that put them in harm's way. And that's one of the critical features of our program as well, that it's not just about the climate and it's not just about the areas in which humans and wildlife come into contact, but the method of how they come into contact. Right. So we do have a behavioral team also that goes to look at qualitative and quantitative data to help determine, you know, if we're concerned about a bat potentially transmitting a disease to humans. Is it when it's already in the restaurant and it's cooked? Is it when the bat is being prepared in the back of the restaurant? Is the transmission route earlier when people are actually catching the bat? And depending on that, and then you have, depending on where the highest risk is, then you have the better chance for making an intervention that will save lives.
Yeah. Now, Dr. G, in some ways that links to you. Can you tell us where we are today in terms of the ways that we can use data science, data analytics, and cognitive computing to answer some of these concerns? I know you've worked in a range of fields, but what can we expect, and where are we going, and why is social information important? Why study social behaviors?
Sure. So I'm personally very optimistic about the direction of using big data to do scientific discovery. So when I say big data, it's not just about the volume of data, but also because the data coming from so many heterogeneous sources. Like you have huge amount of structured literature, right, scientific papers, and you have structured knowledge bases like the cancer pathways, and it can come from multiple data modality, text and image. It can also come from multiple genres. You know, patients may consult social media to ask lots of questions. So I think we are very, we are making lots of progress. So what's most exciting to me from three aspects. First of all, I think we can speed up the scientific discovery dramatically if scientists are willing to collaborate with us. For example, I know in biomedical domain there are more than 300 papers published every day. It's almost impossible to keep track of the progress. And also in this domain, John was telling me that old papers also matter. A lot of hypotheses, you may find them from papers published about 50 years ago, 60 years ago. So how to integrate them and how to discover knowledge graph to help scientists, that's a very big research problem and a promising problem that we can actually have techniques to tackle them. So for example, we recently have worked on a lot of heart disease problems, and we are able to find the most top molecules associated with these interactions between disease and drugs. And also another problem I think a human will have trouble to deal with is uncertainty, because different labs in the world are publishing lots of different papers about the same problem. And, a lab in Harvard may be doing something similar to a lab in China, but they don't know each other's hypothesis. They might be conflicting. But for machine, it's very efficient for us to quickly distill this knowledge and to choose finding and figure out which hypotheses are more credible than others. So I think it's a good intelligent assistant to help scientists to determine the credibility. So, that I think, for example, in our work recently done by our colleagues at RPI and John Doric is on a very unique and ambitious skin cancer problem. It's called I think it's called melanoma, right? No, so there's no certain clue yet how to cure this disease, and it's a very aggressive disease. So what we did was we tried to integrate knowledge bases from unstructured data and unstructured data, and we tried to apply statistical inference to determine which hypothesis or which candidate drug will be the most promising one. And we actually discovered more than 25 candidates, and many of them were very accurate, novel, and that have been tested by other cancers. So I think this is a very exciting direction, and it's really engage human-computer collaboration and human-to-human collaboration. So in terms of the social aspect, I think we need to pay attention, actually a lot of attention to it, is because nowadays patients, they don't trust the doctors alone, right? They want to explore the problem, they want to understand what's happening to my body. So they actually actively consult the web, the social media, they even read the papers to understand what's going on, right? And for them, it's very confusing sometimes because the search engines don't just rank their solution based on credibility. They just look at the redundancy of the information. So I think it's a great opportunity about how we can make the recommendation of information knowledge in a personalized way. So I think that's how advanced data mining algorithm and the natural language processing algorithm can play a role.
Well, you mentioned statistical inference, but you also mentioned taking data from scientific papers and so forth. So would then systems that have a more an agent in the machine, as it were, that has more cognitive capability and artificial intelligence help with this question? So it's not just a statistical inference, but there's machine learning and an ability to look through an enormous amount of data and draw inferences that way from a reasoning perspective.
Yeah, for sure. So I think for most of the state-of-the-art techniques, they are still remain as a black box to both the patients and the scientists. So, but I think we are progressing in the direction that we want to make all the frameworks techniques more transparent. So, for example, if our system, intelligent system, returns top 10 hypotheses, we don't want to throw away the hypothesis in the bottom, because we may receive feedback from scientists and users. They can correct the results, they can provide their own new hypothesis based on their old knowledge. And the framework, the statistical inference framework, should be able to incorporate all those kind of feedback and then make the joint decision. I think that's very important, because we cannot provide 100% reliable confidence estimation yet. But I think that itself is the most exciting research problem.
So John, that naturally turns to you. I mean, how do we turn data into testable hypotheses and from there into action? And so, where are we today and where do we need to go? And maybe you could help us to understand it from the point of view of how it relates to Zika, including some work that was recently done at Rensselaer, or as you've focused on bacterial resistance mechanisms and combating those.
Right. I think that really gets to the heart of the most fundamental question of all of what data science is doing is how do you then translate that into being able to do something with it? So I'm fascinated by what Heng is doing and being able to take data from a variety of different sources, whether it be very structured data that sit in tables, for example, or the unstructured literature and everything in between, including images and things like that. And being able then to parse out potential hypotheses. And at the end of the day, as an experimentalist, we like to be able to develop a hypothesis and be able to test it in a way that advances us in terms of the knowledge we get. At the end of the day, the data that we have doesn't mean anything unless you can draw knowledge from it. So there's a progression that has to occur from the data to the information that you get from that data and how you aggregate that to the ability to actually do get some sort of hypothesis that can be tested either experimentally or potentially with simulations or other data analytic approaches, which allow you then to come in with information at that point or knowledge at that point that could drive a potential solution to a problem. And so let me come full circle to the Zika as an example, as well as the bacterial systems. So as you know, this really began as an observation, the observation that babies were born with a severe brain disease and small brains, small skulls, that obviously is a devastating disease. And it was really a variety of observations. Ultimately, it led to an understanding that there was a viral transmission to that. There had been examples of this over decades, but then it began to become much more severe, certainly down in South America, and now spreading through transmission from individuals as they come up from South America. And one of the things that we just didn't know was when in the development of the human fetus did Zika really come about. And from that you can get information about really the mechanism of the disease. And so this was actually done through some collaborations at Rensselaer with the New York Stem Cell Institute, where they did some in vitro work with stem cells infected with the Zika virus, and then being able to see transcription changes. So when the genes get transcribed over time in the cells, they could then take that information and then use that information to link to the genes to the pathways that are involved in the development of, in this case, neurons and other brain cells, but ultimately can translate that information to the human fetus. And that wasn't really enough because you get a lot of data, and that data is often very hard to understand. So what we were doing, and this was really Kristen Bennett's work in the Department of Mathematical Sciences at Rensselaer, connected to biologists in the Stem Cell Institute, they were able to then take that information and use a visualization tool. And that visualization tool allowed individuals, in this case some of the graduate students involved in the work, to actually look at connections between various genes that are expressed at different times following the Zika exposure. And then they began to take that information together with the data analytics of how these genes, or when are they turned on, when are they turned off, and things like that, ultimately being able to identify specific pathways and times in development that those pathways were impacted by the Zika virus. Now, does that lead to a cure? No. But it does lead to a better understanding of what the mechanism is. And once you have a better understanding of the mechanism, you can then begin to develop either better diagnostics, so that you can test whether or not something has happened in the very early weeks of fetal development, or you can ultimately potentially develop some sort of treatment. Now on the bacterial side, As you know, one of the biggest problems we have in our developed world as well as in the developing world is the gain resistance that bacteria have to antibiotics. So antibiotics were the panacea from the 20th century, being able to cure people from deadly diseases that up until that time, there was very little that can be done. But today, in the US alone, it's the 4th or 5th leading cause of death, hospital-acquired and other drug-resistant bacterial infections, the 4th or 5th leading cause of preventable death in the US. And the World Health Organization has even come out saying that we're moving back to the early 20th century, before antibiotics were actually developed, in terms of our ability to fight infectious disease. So again, data comes in here because we had known from back in the 1950s, 60s, that nature actually has a variety of antibiotic-like systems that are not small molecules, but in fact are enzymes. And these enzymes are co-evolved with the bacteria that they actually target. So in fact, the bacteria have a very difficult time gaining resistance against them. And these enzymes can be used selectively against a specific bacterium. They're going to be so specific that they even can attack specific species within the genus of a bacterium. And that avoids the broad spectrum antibiotics that have been really the cause of the antibiotic resistance, because they wipe out all the microbiome in your body, or a large part of that, leaving the pathogens alone to get resistant and then go and take over and lead to the various diseases. So in this case, what we simply did was, there's a huge amount of genomic data that's now available. In fact, even data from metagenomes of organisms that are not even culturable. And from that, we can very quickly search using a variety of very high-powered tools and using high-performance computing capabilities as well, to be able to identify purported genes that might in fact consist of a code for these enzymes that could very selectively kill bacteria. And by doing that, we ended up developing and identifying a range of new types of enzymes, enzymes that actually come from bacteriophages, which are the viruses of the bacteria that they infect, that can become quite useful to kill various bacteria, anything from MRSA to Listeria to Bacillus. And to do that in the exclusion of killing any other bacterium, which of course means that you're targeting the bad bacterium that you want to get rid of and not hurting the good bacteria that you want to keep.
So if I've heard you right, we know that 3/4 of human diseases of certain types have zoonotic origin, meaning they start with animals. And we know that those pathogens can jump species. We know that in the process, and this is just an add-on, I was studying the great 1918 influenza, that certain kinds of viruses also mutate more easily, and each time they can jump, they can mutate. We know that the problems are both scientific or medically rooted, but they're behaviorally rooted as well. We also know that now, with new approaches to data analysis, data analytics, that we're able to take data from multiple sources, and increasingly in both structured and unstructured form, and from multiple types of forms, not just text. And we're able to begin to marry that with things we learn about behaviors to create hypotheses to explain certain phenomena. And so then what you've been saying, Dr. Dordick, is that one can use those kinds of techniques and marry them with the data that has come out of work people have done, in this case in genomics and so on, to be able to not only create testable hypotheses, but then to think of therapeutic pathways that can help us to address at least certain classes of diseases. Have I captured this in the right way?
I think very well, yeah.
I'm a physicist. And so with that, I'm going to ask you, John, to try to carry on the conversation, because I think it's important that people kind of understand, you know, what happens on the front lines of diseases, and what do we, you know, really, what are we able to do? And how do we bring people together in ways to make more progress across the disciplines? So
Okay.
I'm going to leave it to the experts.
Yeah, thank you very much. So, Dr. Murray, maybe you can give us an idea of really the sources of the data that you're actually dealing with. And I know they're very different in many cases. And when you deal with data, and you'll probably have to deal with this too, how do you know they're reliable, if the data is reliable, where you get it from, where it comes from, how it was obtained, and experimentally you have to obviously look at what that means from a data reliability standpoint. And then from that, how do you really extract the knowledge that you need in order to do the things you do to try to address issues in the zoonotic transmission of diseases and ways to prevent that?
The first thing I would say about addressing issues and collecting the data and interpreting is that teamwork cannot be underestimated.
Yep.
And that's something, as a wildlife veterinarian, that I have a lot of expertise in anesthetizing different species and actually getting the biological samples and getting them to the lab. On our emerging pandemic threats program, we actually have a data management team, and we have a modeling team, and we have a surveillance team, and a capacity building team. So in each of those, we have experts in each area, and then we also have, you know, broader team meetings, at which point that monthly, where everybody reports, so we can learn from each other. And so I hope I do it justice, but in terms of the people who are actually collecting the data, we have we have all the countries that we work in have country coordinators, and everybody who is collecting the data has a specific check sheet to go through, from what data was collected in what area, what animal, what location to which people. So we have a high degree of confidence in the data that we're currently utilizing, because it's our own team that's collecting it, and it's an iterative process in which we teach, and then we teach again, and then we're present as our team continues, and then we verify the data. So I would say that I personally am not a good data manager, but we rely on it heavily, and so in that case we trust the experts in terms of to make sure the data is actually accurate.
Well, how do you deal with, you just brought up a very broad team.
Right.
With very different expertise.
Yeah.
From data and computational to experimentalists and so forth. How do you get them to communicate with each other in a way that will enable them to listen to each other, and that you can then, from that, figure out what to do next?
I would say that the PI of the entire program is a woman, Dr. Jonna Mazette from UC Davis, and I think that she is brilliant, and I watch and I pay attention to her a lot, because she does find a way to figure out which teams we need to empower them separately, but also to make sure we're all communicating. So I would love to take credit for that, and I would love to, but it's not me. I watch someone else who's been able to very much lead the team as a whole through consistent communication, and then a lot of feedback.
Okay, great. So Heng, maybe you can go down to the basics about how do you actually develop the hypothesis or the hypotheses that you can obtain from mining all this data in very different forms. The 300 new papers a day, the massive number of papers that have been published in multiple different fields, many of which are unrelated to what people typically search for, and yet may be very relevant from studying a complex a problem, whether it's a disease or some other target and outcome that you're looking for. And how do you actually do the mining of these structured and unstructured systems?
Yeah, so we built sophisticated pipeline, but basically the idea is we try to narrow down the information by document retrieval, that kind of technique first. So we know this subset of documents are things we want to focus on. Then we zoom in to look at how can we build comprehensive knowledge graphs by aggregating information across these documents. But that just gives us some surface information. And then we try to build statistical models. For example, we have state-of-the-art truth finding model in which we actually put 3 dimensions. So we have the user or information provider dimension. That's what you mentioned, things like expertise. So we have automatic way to quantify the expertise level of the information provider. Right? So like this researcher published many papers on this particular topic, then we promote this person's credibility. And then we link that dimension to the second dimension, which is the knowledge graph dimension. So, and then the problem is many of the data mining and the natural language processing techniques were developed for general domain, like news articles, right? So when we adapt them to this particular biomedical domain or any other domain like chemistry or engineering domain, we will always have the information loss problem, right? Because the systems are not perfect. So we usually allow the systems to provide the confidence. So it was humbly say, even if I'm not very sure about my hypothesis, this is my confidence value, 0.3. And then we link the 3 dimensions together and we do propagation. So it's like although this hypothesis itself from the system generation point of view is not so high, but it's coming from a credible researcher like Suzan, then we promote that hypothesis. So it's a back and forth propagation model across the dimensions.
But if you, let me play devil's advocate and say, let's say you have a young faculty member, and that person hasn't built up credibility yet, right?
Yeah.
So, but they may have come up with one hell of a hypothesis. So then what happens?
Yeah, that's an interesting question. I think the credibility is not just based on majority voting, like how many credits you have aggregated from the past. We also try to find, for example, novelty detection, right? So if the hypothesis from this particular paper is so unique, then it will become an anomaly point when we construct a graph, right? And then the model probably won't say this is best one, but it will at least tell the scientist, this is something you should pay attention to because it's so unique. So we incorporate multiple criteria into the model.
So why don't we open up the questions to the audience and be happy to take any questions you have, any comments, and see where we go from there. So any takers?
This is more for Heng. The methodology that you just said, where does the use of ontology sit in that framework? Is that different from the approach that you just said, or is that part of that approach?
It's actually in the intermediate step of this approach. So when we construct knowledge graph, it's not just from the unstructured data. So we build a baseline from the scientific papers, and then we link the concepts to ontologies. For example, when I was talking about the heart disease problem, we actually enrich the knowledge graph by linking each concept to 300 ontologies. So we are trying to simulate how human would do this, right? Like when you cannot understand or disambiguate the concept in the paper, you usually will refer to the ontology and then try to read the description and then try to figure out what that concept is. So we let the machine to simulate that procedure. So the input to the machine, to the program, is not just the paper itself, it's actually enriched paper. So each concept has the properties and the description we automatically discover from ontology. So it's a very important step, actually.
Thank you.
Yeah, in the process.
Any other questions? Yes.
Hi, I wonder if you could give us a prognosis for antibiotic resistance and our capacity to pull back from the early 20th century. And how bad is it going to get before it gets better?
Well, I think there are a number of points to this that worry me. We know that we have tens of thousands of people die a year from a variety of different types of hospital-acquired infections alone in the U.S. That's not a huge number. Mm-hmm. Obviously, the number should be zero, but that's never going to be the case. But we are also having that occur at the same time that pharmaceutical companies are generating fewer and fewer new antibiotics, partly because the low-hanging fruit is gone and the likelihood of resistance is high to happen very quickly, so there's not much of a lifetime left of the drug once it gets through approval. And there's also significant issues with respect to toxicity they worry about, so approval becomes a complication. But at the same time, pharmaceutical companies prefer to work more on the chronic diseases. When an antibiotic, if you do it right, you take it for maybe a week to 10 days, you're done, that's it. Whereas, if you take a statin, it may be for the rest of your life. So there's a confluence of all that coming together, but there's also issues, environmental issues. So the overuse of antibiotics, of course, we know. People go in, they have a cold, and they feel they need an antibiotic. They convince their doctor they need it. It won't do a thing to a viral infection. And yet antibiotics make its way into the water system from that, from sanitation and so forth. Even worse is antibiotic use in the food industry, which is used in a number of ways. One, to maintain infection from overtaking close-knit animals like chickens and so forth. Mm-hmm. But the other one is to fatten up cows and pigs, because you don't want the bacteria in the gut eating any of the carbon source, any of the food, and turning it into gas, in methane. They want to convert it into body mass. And then that makes its way into the fields and into the water system and so forth. You could take any water in the U.S. and take a glass of water, and you can measure the antibiotics in there right now. And so that is just breeding very quickly, because bacteria are quite smart, because they have evolution behind them, and they can mutate extremely quickly, and they do. And so the problem is exponential, and that's what I worry about the most. So today we can hold it down a bit, except in certain cases, in certain hospitals, it's a problem. But it doesn't take much to explode from there, and that's what worries me the most.
I was looking for some reassurance, but I guess I didn't get any.
Well, I mean, we're not here to scare you, although, quite frankly, there are a number of things that could be done right. We can stop using certain antibiotics just because people feel they need it. The other thing is we do need to look at different ways of combating the disease. So nature makes antibiotics. So most antibiotics are natural products, and it's made by given bacteria to fight off another bacteria. But the environment that those bacteria are in are very well defined. And so it turned out, yes, we can use it very broad spectrum. But in the end, that's not what nature designed it for. So we can go back to what nature did design. So what nature did design are viruses of bacteria that kill very specific bacteria. And so that's another way to go. However, that's not a popular way to go yet. It could be. A lot more research is needed to be able to do that. And so if public health, if the federal agencies, not just in the US but around the world, take this more seriously, then I think we're going to see a paradigm shift in what kinds of therapies we might be able to have down the road. Because at the end of the day, what I worry about the most is not just the bacterial infections that we know of, but it's the hidden bacterial infections in our body that may be very much responsible for heart disease, cancer, and so forth. And it's only beginning to come out. And then the healthy versus unhealthy microbiome, which is also a very interesting thing to consider from a research perspective, that can have a huge impact in the way we live our lives and the diseases we look to avoid. Yes.
What's going to be your recommendation, each of you panelists, that in terms of global health governance point of view, that how can you use big data to really, how can I say, manage that pandemic things in the future?
Yeah, I'll start with that one. That from my perspective, I think that some of the things that what governments can do right now is to really embrace the One Health concept more. The idea that the survival of one species is very interdependent upon the survival of all species. So if we're trying to look at the health and survival of humans, it can't be in a vacuum, and not and it's very much interrelated. The emerging infectious diseases is very much interdependent upon the wildlife. So the countries that are really embracing the One Health platform,
Global health protocol,
yeah.
global health governance, because this is not only for the one or two countries, but global governance.
Right.
For the data, big data.
Yeah, it's right. So we need to have many different experts working together. From my perspective, again, the physicians and the veterinarians, and that data needs to be collected up front, before the next outbreak. So surveillance team getting an understanding of what's going on now, as well as the building of laboratory capacity, so there's a much quicker response time. So if that's something that I think governments can do now to really invest in disease surveillance, and again the idea of the overlap between human and veterinary health.
Yes, I think it will be really, really helpful if government can set up some more funding opportunities for interdisciplinary collaboration, not just within, for example, U.S. government, but also across different countries. For example, if we want to deal with any disease in West Africa, we probably have to have easy access to any data, right? They probably have some news articles reporting this disease, but in a different language, a foreign language. So it would be really great if we have this nice infrastructure that can really tightly couple computer scientists who are experts in big data mining and domain experts, medical domain experts who can interpret the results. So I think we are making good progress. Like in US, we have not only have NIH, they have the big data program. They also have like DARPA recently set up a program called Big Mechanism. They are trying to predict or reconstruct the cancer pathways from scientific literature. So I think US is still pioneering that direction, but I think data sharing is a big problem. So if different countries can do close collaboration, build an international platform, I think that would be really wonderful.
And I think also, just before John's about to speak, I think that for the three of us, the more that we can collect data, the more that we can develop systems to analyze that data, and the more that we can develop the interventions based on the information that each of our teams provides. I think that would only aid our success.
Yeah, and I guess I would add to that by saying that what we're really looking at from the governments are commitments to fund the basic research. These are targets that we've got to go after, but we understand too little. So as a result, when we get all this data, it may tell us something, but we don't understand what it really means. But it tells us to do X, Y, and Z. But without understanding the biology, without understanding the nature of the data itself, without being able to develop the unique algorithms or maybe the cognitive systems that would enable us to understand the data better, and ultimately be able to do the basic research experimentation, we're not going to be able to attack the problem in a way that's smart. We're going to attack it little bits here and there, and we're always going to be running behind what nature can do, because again, nature is pretty smart. So maybe I'll just wrap up, because we do have to move on. I hope you've heard a little bit about things that won't scare you, but it may get you to think about that it is important, and that it is a complex problem, and that in the last few years, this confluence of data science in its broadest term, together with experimentation, and that experimentation involves not just being in a laboratory, but being really within the entire world itself, its whole environment, multi-species and so forth, is really just the tip of the iceberg of what is ultimately going to happen. and that this is an exciting time that we now have some of these tools to be able to do things, and hopefully we'll be able to take those tools to the next level soon and really make the quality of life of all of us improve. So thank you very much for attending.
Thank you.